Hello, everyone, and welcome to today's webinar titled HIV-1 Genotyping Targeting Integrase, Perspectives on Surveillance of Antiretroviral Drug Resistance. This webinar is part of the 13th Annual Clinical Diagnostics and Research Virtual Event, and I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Thermo Fisher Scientific. For more information about Thermo Fisher Scientific, go to www.thermofisher.com. Now let's get started. Before we begin, I want to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our new live chat feature during this presentation. You can find that live chat located on the left of your screen. You can also participate by submitting as many questions as you'd like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. You will also notice that the speakers have provided a survey located on the right of the abstract. This survey will be available during both the live session and on the on-demand period. Now, if you have any trouble seeing or hearing this presentation, just simply click on that Help Desk button located at the top of your screen within the navigation bar or from the lobby. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on that continuing education credits link located at the abstract tab from the menu at the left of your screen and follow the process to obtain those credits. I now present today's speakers, Dr. Kim Stegen, a senior medical scientist at Charlotte Maxey Johannesburg Academic Hospital, and Dr. Natasha Wood, co-founder and chief science officer at Hyrax Biosciences, and Dr. Steve Williams, an R&D leader at capillary electrophoresis at Thermo Fisher Scientific. For a complete biography of our speakers, please visit the presenter tab from the menu at the left of your screen. Dr. Stegen, Dr. Wood, Dr. Williams, you may now begin your presentation.
I would like to thank Thermo Fisher for inviting me to present today on the global status of HIV drug resistance. By 2020, about 37.7 million people were estimated to be living with HIV, including 20.6 million people from Eastern and Southern Africa, which represents the majority of the epidemic. Antiretroviral rollout has started in 2000, and by 2020, about 27 million people were on ART, which is a threefold increase since 2010. The massive rollout of antiretroviral treatments has also significantly had an impact on the decline of AIDS-related deaths. In 2016, the UN set up some goals in order to have as many people living with HIV to know their status, of those that know their status to be on treatment, and of those that are on treatment, the majority being virally suppressed. Although those targets were not met by the end of 2020, we were not far off. 84% of people living with HIV know their status. Of those that know that are HIV positive, 87% are on treatment. And of those that are on treatment, 90% are virologically suppressed. HIV is a virus that has a very high genetic diversity. This diversity is driven by the production of 1 to 10 billion virus particles per day, and in addition to the reverse transcription process being prone to error. The reverse transcriptase enzyme does not have a proofreading mechanism to correct for the mistakes, and therefore we expect about 1 random mutation per new provirus. The higher the levels of viral replications, the greater the risk for the development of drug resistance, as more replication means more mistakes are being made. Virological suppression is not only important to reduce the risk of transmission, but also to reduce the risk of resistance. Therefore, we need efficacious antiretroviral treatment. When we look for efficacious antiretroviral treatment, we look for drugs that have a very high viral potency, have a very high genetic barrier to resistance, and in addition, good safety profiles, limited drug-drug interactions, and the possibility of co-formulation between different drugs increase the adherence in patients and therefore increases increases viral suppression. In this figure, the HIV life cycle is represented with the different steps in the life cycle, and it's indicated which different classes inhibit the different steps in the life cycle. The most commonly used drug classes are those that inhibit the reverse transcriptase step, including the NRTI and the NNRTI drugs, as well as the protease inhibitors. And more recently, the integrase inhibitors have been included as well. Initially, the integrase inhibitors were mainly focused in resource-limited settings to treat patients on third-line treatments, whereas with the introduction of dolutegravir, there has been a shift of using integrase inhibitors in first-line treatments. However, even though some of the newer drugs have a very high genetic barrier and are very good drugs, all available drugs, including the newer classes, are at risk of becoming partially or fully inactive due to the emergence of drug-resistant variants. HIV drug resistance can reduce the effectiveness of antiretroviral treatment, not only on an individual patient level, but in a global perspective as well. Therefore, it is important to monitor HIV drug resistance, especially in regions where HIV drug resistance testing is not available at patient level. The outcome of these surveillance efforts can then inform public health treatment guidelines. The World Health Organization has set up an HIV drug resistance strategy, and they report on a regular basis on the outcomes of countries that have performed drug resistance surveillance. So in the next couple of slides, I will touch on a couple of the results that were published in the most recent 2021 drug resistance reports. There are four main pillars in the drug resistance strategy from WHO. The first one is focusing on pre-treatment drug resistance in adults initiating or reinitiating first-line treatment. The second one is pre-treatment drug resistance in ART-naive infants who are newly diagnosed with HIV. Number 3 is acquired drug resistance in adults, children, and adolescents. And finally, surveillance of patients who are on pre-exposure prophylaxis who are diagnosed with HIV. I will discuss some of the results for the first 3 pillars. I will, however, not touch on results from the last pillar. Starting with pre-treatment drug resistance in adults initiating or reinitiating first-line treatments. 21 out of 30 surveys reported first-generation NNRTI pre-treatment drug resistance levels above 10%. This is indicated on the graph on the right with the vertical red line, which indicates the 10% cutoff. All countries that reported than 10% do have dots on the right-hand side of the bar. So you can see that NNRTI drug resistance is quite significant in most countries in patients initiating or reinitiating treatment. Overall, the prevalence of first-generation NNRTI pre-treatment drug resistance was 3 times higher among people reporting previous ART exposure, being on average 24%, compared to those who are truly ART naive, being 7%. The prevalence of pre-treatment drug resistance overall is still driven by NNRTI resistance, which is indicated in the red box. On the other hand, NRTI resistance in the amber box remains relatively low, and if we look at PI pre-treatment drug resistance, it's even lower, as for the same with second-generation integrase resistance. Based on these results, there has been a change in many treatment guidelines, including the WHO guidelines, where the preferred first-line treatment regimen has been changed from an NNRTI-based regimen to now a combination of tenofovir, 3TC, and dolutegravir, which is offered in a fixed-dose combination, also referred to as TLD. Moving on to pre-treatment drug resistance in ART-naive infants who are newly diagnosed with HIV. There were surveys conducted between 2012 and 2020 in about 10 countries. And NNRTI resistance was observed in over 70% in some of the countries. Overall, across all the countries, at least half of the kids born HIV positive had at least one drug resistance mutation. Although the level of NRTI resistance was a bit lower, it still exceeded above 10% in most of the surveys conducted, meaning that there is quite a bit of transmitted resistance between mom and child. Based on these guidelines, based on these results, the guidelines have changed once again, whereby WHO initially recommended a PI-based regimen, moving on from an NNRTI-based regimen, but now with the introduction of dolutegravir, the preferred regimen is actually a dolutegravir-based regimen. Finally, moving on to the acquired drug resistance in adults, children, and adolescents. So this is when surveys are being done in patients that are on treatment but are failing at the time of resistance testing. Surveys are usually conducted at 12 months and then at 24 months of treatment. In this graph, it is obvious that most patients failing first-line NNRTI-based treatment have a very high prevalence of resistance, in some countries over 90%. When we look at surveys done at patients that have been on treatment for 48 months, the level of resistance drops slightly, but there is still a very common presence of resistance in these patients. So based on these results, it is once again obvious that NNRTI-based ART has a very low genetic barrier to resistance, meaning that many patients failing these regimens present with drug resistance. Therefore, the second-line treatment guidelines have now been changed. For adults currently failing non-DTG-based ART, they should be switched to a DTG-based regimen with an optimized NNRTI backbone. And in addition, adults that are currently failing a first-line DTG-based regimen, they should be switched to a boosted PI-based regimen. Among the surveys that were conducted and that were reported in the WHO report, only 10 out of 35 countries included integrase resistance surveillance. In these surveys, less than 0.4% of drug resistance was detected, which is extremely low. We do, however, need to take into account that the rollout of DTG so it is very common that we don't see a lot of resistance. Therefore, WHO recommends to include INSTI resistance testing for all new surveillance activities going forward. Switching gears a little bit now, so we're now no longer talking about the WHO reports, but because there is so limited information available on integrase resistance, I thought it would be informative to also look at what is happening in the Western world. In this study in the US, they looked at 28 jurisdictions between 2014 and 2018, included more than 50,000 people, of which almost 20% had at least one drug resistance mutation. Despite that the rollout of integrase inhibitors have been much longer compared to low and middle income countries, they still only see a 0.8% prevalence of integrase resistance. And the resistance in pretreatment patients is still driven by NNRTI resistance. In a recent review, they looked at acquired resistance of patients failing DTG-based treatments, and they found that the risk factors for DTG resistance were mainly poor treatment adherence, drug interactions, and HIV disease factors such as high baseline viral loads and active opportunistic infections. In the review, they only included 15 reported cases of dolutegravir resistance because dolutegravir resistance is so rare. Of those 15, 10 had either suboptimal DTG levels or poor adherence. 3 patients had advanced HIV and severe active infections, and 4 treatment-naive and 5 treatment-experienced people had a baseline viral load of over 100,000 copies. There is, however, a concern that the limited access of viral load and genotypic testing in low and middle income countries may influence the proportion of people with virological failure and therefore the emergence of integrase resistance. In Canada, a surveillance was done between 2009 and 2016, including close to 10,000 individuals, and they found that the level of protease and reverse transcriptase resistance decreased significantly from 337 cases per 1,000 treated individuals in 2009 to 285 per 1,000 treated individuals in 2016. On the other hand, the integrase resistance increased significantly but remained very low. It went from 1 per 1,000 treated individuals in 2009 up to 7 per 1,000 treated individuals in 2016. Of the 64 individuals that were newly identified as having integrase resistance, the majority of them were driven by exposure to raltegravir, which is a first-generation integrase drug, whereas there were only 3 cases of new integrase resistance mutations for patients with DTG treatments. So to date, most surveillance efforts were focused on the protease and reverse transcriptase gene, but with the massive rollout of dolutegravir and possibly other integrase inhibitors, WHO recommends to include integrase resistance in any new surveillance activities. In conclusion, HIV drug resistance surveillance has informed policymakers on the best treatment regimens in certain populations, especially in regions where a public health approach is required. As countries scale up the transition to integrase-based antiretroviral treatment, surveillance of the integrase gene, especially in low and middle-income countries, is required. Thank you for your attention.
Thank you very much, Dr. Stegen, for the informative presentation. And hi, everyone. Thank you for joining. As I've been introduced, my name is Natasha, and I'm from Hyrax Biosciences, which is a company that develops software for DNA sequence analysis. And today, specifically, I'll be talking to you about our X-Type Sanger HIV product, which is a platform for HIV drug resistance analysis and surveillance monitoring. So just as a presentation overview, I'll first provide you with a brief introduction of the X-Type workflow. I'll then jump into a series of screenshots to show you exactly how X-Type works. And then finally, I will conclude on how Thermo Fisher and Hyrax Biosciences are supporting this global drive for increased surveillance. So, as I've said, X-Type Sanger is an online platform which you can access at the URL at the top left, and it is an automated HIV drug resistance reporting platform for Sanger sequencing data. And to place it more in the context of HIV genotyping, if you think of HIV genotyping as a 3-step process, the first step is to do sample prep, where you extract your DNA or RNA. The second step is the sequencing, where you generate your data. And the final step here is the analysis, where you actually do the interpretation and the analysis of the data. And this is where X-Type comes in. And we really think about X-Type as an out-of-the-box solution, which consolidates the global best practices for HIV Sanger data analysis, specifically for HIV. So what X-Type does is base calling and editing. It does consensus sequence generation, quality control analysis, drug resistance reporting for surveillance. The results are available for download in multiple formats, and you can share your results between peers within an organization. And if you're wondering what the underlying programs and software are that power X-Type, we've really consolidated the global best practices. So on the left over here, you can see your AB1 files, which come from the sequencing instrument. And in the next image, we have licensed the University of British Columbia's Recall software to do chromatogram editing and review. So when you see this online on the user interface, you are actually using the Recall software, which you may be familiar with. Then finally, on the next image, the Stanford HIV Drug Resistance Database, we have implemented this algorithm in our software as well. And just a note on this, when Stanford updates the drug resistance database and the drug calls and resistance scoring, we update it on our side within 48 hours. So you're always running the most up-to-date software on X-Type. And then finally, on the right, you can see that you get your drug resistance reports and drug calls for surveillance and research. And we also provide these quality control and surveillance monitoring results, which have been laid out by the WHO in this document shown over here, which consolidates all of these best practices. So that, in a nutshell, is an introduction to Exotype, and I'll now take you through a series of screenshots. So once you have logged in and created an account, you will access the dashboard over here. To run your data, you create a new job or a new analysis, and you go through a series of steps. Over here, I've shown you the first step, which is just to create the job. And you can see at the bottom left, you need to select if you're running the HIV genotyping kit with Integrase or without Integrase. And these are provided by Thermo Fisher. So once you've gone through the series of steps, which you can see in the tabs on the top, so job details, select data, all the way to review and submit, which is the final step. Once you click this, you will then be taken to a results page or a pre-results page. And here you can see on the left, in orange, you've got all the samples that you've uploaded. And for each of these samples, you then have an associated chromatogram file. For each sample, if you've run both protease and reverse transcriptase, as well as Integrase, you will have a separate chromatogram for each of these gene regions. And as you are more familiar with, and I am, I'm sure, you will be able to do your chromatogram editing and review for each of these gene regions and each sample. Once you've completed these steps, you will then have your results. And the results include a quality control overview. And over here, you can see the results of the sequencing control analysis. You also then have sample-specific results, which include the sample warnings, as well as drug calls and drug scores for surveillance. And then finally, you have a series of downloads that are available for downstream analysis, which include your consensus sequences in different formats, as well as a detailed summary file, which is a tab-separated file that you can open in Excel. And this file is particularly useful for your surveillance, large-scale surveillance studies. So I know I've just shown you a few screenshots here, but I would like to encourage you to please get in touch if you would like a demo or a training session, and you can get in touch at sanger@hyrexbio.com. So please don't hesitate to get in touch for a more in-depth overview. And then just to tie it back into the surveillance studies, there are several indicators for surveillance studies, and one of these are the sequence similarities. So if two viral sequences are too similar to one another, this may indicate specific transmission chains. So reporting on sequence similarity is very important to monitor your transmission dynamics. To show you how Exotype does this, in the right-hand side image over here, you can see in orange that 4 samples have been flagged as too similar to one another. So D8, D6, D9, and D12 are too similar to one another. There are too few differences between their nucleotides. And if you then plot this on a phylogenetic tree, you can see that these 4 samples cluster on a single branch, which indicates that they may have been transmitted as a cluster. So before I hand over to Dr. Williams, who will take you through the sample prep and the sequencing steps, I'd just like to conclude that as HIV genotyping scales across the world, these kinds of streamlined laboratory methods and sequence analysis tools are really key to facilitate the ongoing surveillance of drug resistance mutations. And this is especially in light of Dr. Stegen's presentation, which presented the WHO drug resistance report, and it highlights the challenges of implementing these national surveillance plans. And then finally, again, I would just like to encourage you to get in touch for a demo or a training session if you are interested. And thank you very much for your attention.
Hi, thanks for joining me today. I'm Steve Williams. I lead the assay development group within the clinical testing solutions business at Thermo Fisher Scientific. And today I'd like to describe the design and the performance of the Applied Biosystems HIV-1 Genotyping Kit with Integrase that is able to detect HIV drug resistance mutations relevant to the most widely used drug classes across a broad range of viral subtypes. As you may know, Thermo Fisher launched the first iteration of this product, the HIV-1 Genotyping Kit, in 2017, and it's been widely used to help survey drug resistance for protease and reverse transcriptase inhibitors in low to middle income settings. The new genotyping kit that we're introducing retains the same excellent performance characteristics of the original kit, but adds additional sequencing of a region of the integrase gene as the use of the integrase inhibitors expands in low to middle income countries. So specifically, the HIV-1 genotyping kit with integrase detects resistance mutations in protease, reverse transcriptase, and integrase. It is a cost-effective solution and has broad coverage of all of the HIV-1 group M subtypes, including the circulating recombinant forms of the virus. The kit is compatible with plasma and dried blood spot sample specimens, and it has a scalable workflow that can deliver a drug resistance result in about 14 hours, all the way from RNA extraction through a drug resistance report. And that workflow represents less than 3 hours of total hands-on time. The kit comes with pre-mixed reagents to minimize the number of pipetting steps, and includes a number of controls in case the need for troubleshooting. And the analysis of the Sanger sequencing trace reactions can be performed on a series of different genetic analyzers that represent different throughput requirements. And then finally, as Natasha explained in the previous slide, we have a easy-to-use software to interpret the Sanger traces and to identify the drug resistance mutations. So the assay workflow is shown in this slide, beginning with the specimen processing to extract the viral RNA. After RNA extraction, there is a RT-PCR step to amplify the virus, followed by a second amplification step. That's a nested PCR reaction, and then Sanger sequencing of the drug resistance regions and interpretation of the Sanger sequencing traces is performed. As mentioned previously, the end-to-end process takes about 14 hours with about 3 hours of total hands-on time. The RNA extraction can be performed using the MagMax viral pathogen kit from Thermo Fisher, although any extraction method yielding high-quality RNA is acceptable. The MagMax protocol has been optimized for both plasma and DBS samples. And the genotyping kit itself consists of two different modules. The first module is an amplification module that does RT-PCR, followed by nested PCR. And the second module is the sequencing module. Importantly, the first amplification step can amplify all three regions of the virus, protease, reverse transcriptase, and integrase, in a single step. So there's no need to split the extracted RNA sample into two different amplification reactions. The second nested amplification step does require splitting the PCR reaction into protease and RT nested PCR and also integrase nested PCR. And then the second module in the kit consists of all of the sequencing mixes that are required to sequence the amplicons prior to running capillary electrophoresis to separate the Sanger fragments and call bases. And then the final step in the workflow is the assembly of the individual sequences into a consensus sequence by the Exotype software and calling of the presence of any drug resistance mutations. Drilling down in a little more detail, the HIV-1 genotyping kit amplifies the protease, reverse transcriptase, and integrase regions, which can predict drug resistance for nucleoside reverse transcriptase inhibitors, for non-nucleoside reverse transcriptase inhibitors, protease inhibitors, and integrase inhibitors. In the PR region, codons 6 to 99 are amplified. In the RT region, we amplify codons 1 through 251. And in the integrase region, codons 1 through 288 are amplified. So the first step, as I mentioned, of the amplification is a multiplex RT-PCR, where both protease, RT, and integrase are amplified. And this produces two different fragments, which are further amplified in a nested PCR protocol. For protease and RT, we produce a 1.1 kilobase fragment, and for integrase, it's a 1 kilobase fragment. So following amplification, the success of the amplification is confirmed by running a gel and looking for a band of the correct size. At this point, if the amplification is successful, you proceed on to the sequencing step of the workflow. The sequencing is performed with, in the case of protease and RT, 6 different sequencing primers to cover the entire length of the nested amplicon. These are 3 forward sequencing primers and 3 reverse primers. And in the case of integrase, 4 different sequencing primers are used to cover the amplicon length, 2 forward primers and 2 reverse primers. The result of the sequencing is that for any given nucleotide in either of the amplicons, you have at least 2x coverage, both a forward and a reverse read for each nucleotide. And following sequencing, the reads are assembled by the Exatype software, and the consensus sequence checked against the Stanford HIV drug resistance database. So the performance of the HIV-1 genotyping kit was validated in collaboration with the CDC in Atlanta with our colleagues. And as part of the validation, the performance was assessed according to the WHO validation criteria, where we looked at subtype coverage, accuracy, amplification sensitivity, precision, and reproducibility. So briefly, this validation procedure starts with either 200 microliters of plasma or 1 to 2 dry blood spot samples. RNA extraction is performed, and then 10 microliters of extracted RNA is input into this multiplex RT-PCR reaction to amplify the 3 different drug resistance regions of the virus. The RT-PCR reaction, as we've discussed, is split into 2 different nested amplification steps, one to amplify protease and RT, a second to amplify integrase. And then finally, the nested amplicons are sequenced by Sanger sequencing and run out on the CE instrument. So to demonstrate the broad subtype coverage, samples with known subtypes were analyzed in both plasma and DBS formats. In a total of 139 samples were analyzed that were split between 39 clinical research samples and 59 analytical samples. The clinical research samples came from Nigeria, Kenya, and Haiti, and the analytical samples were a mixture of either external quality assessment samples from proficiency panels provided by the CDC, viral isolates, or cultured integrase clones from Stanford. And as you can see there from the table in the slide, there was successful amplification seen across all of the major group M HIV-1 subtypes, including the CRS and recombinant subtypes as well. Next, we looked at the assay accuracy. So the accuracy was determined for both plasma and DBS samples across a wide range of viral load, all the way from the limit of detection of the assay up to quite high viral loads, up to as high as 10 to the 6 copies per mil. The accuracy was determined by comparing data against reference sequences from 3 different sources. Firstly, for the clinical samples, clinical research samples that we looked at, the sample was compared against the sequence data from the original Thermo Fisher protease RT kit, or in the case of the integrase region, against the ViraSeq integrase assay. For the proficiency samples that were looked at, the consensus sequence from the proficiency exercise was used as ground truth, and for the isolates, data from Stanford University, sequence data from Stanford University was used. So the conclusion from the accuracy study is that for both plasma and DBS samples, more than 90% of the comparisons between the test result, the result run with the new HIV-1 genotyping kit, and the reference sample were at least 98% identical. So very high concordance with the ground truth in this case. Next, we examined the assay amplification and the amplification sensitivity. And this was measured by the presence of a band on the gel for the nested PCR product. It was determined for 2 different groups of plasma and DBS samples. The first group had a viral load of greater than 5,000 copies per ml, and the second group had a viral load between 1,000 and 5,000 copies per ml. And for the second group, this was considered at or close to the LOD of the assay. For the plasma samples, we found that more than 90% of the specimens and replicates were positive when the viral load was between 1,000 and 5,000 copies per ml, and more than 95% of the samples were positive, i.e. they produced a band on the gel when the viral load was over 5,000 copies per ml. For the DBS samples, more than 85% of the samples were positive when the viral load was close or at to the limit of detection between 1,000 and 5,000 copies per ml. And at the slightly higher viral loads above 5,000 copies per ml, more than 90% of the samples produced a band on the gel. So good assay sensitivity for both plasma and DBS samples. We also looked at the assay precision and reproducibility again, across a wide range of viral loads, all the way from, in the case of the precision study, 5,000 copies per ml, and the case of the reproducibility study, 2,000 copies per ml, up to quite high viral loads. For the precision studies, 3 sample replicates were extracted and tested together by 1 laboratory scientist. And for the reproducibility studies, 3 plasma samples at different viral loads were run with 5 replicates on different days by 2 lab scientists. And then pairwise comparisons at the nucleotide level were performed to assess the reproducibility and precision. And in all cases, the pairwise comparisons at the nucleotide level were very, very good, greater than 99% across all of the viral loads examined. So in conclusion, the HIV-1 genotyping kit with Integrase meets all of the WHO HIV-DR assay performance criteria for surveillance. It has broad range of subtype coverage for both plasma and DBS samples, has excellent specificity and a sensitivity of 1,000 copies per ml for plasma samples and 2,000 copies per ml for DBS samples. A range of different capillary electrophoresis instruments can be used depending on throughput needs to analyze the Sanger sequencing reactions, and the workflow enables the user to go from sample specimen to cold drug resistance mutation in less than 14 hours with about 3 hours of total hands-on time. And the kit is configured with premixed primers, and all of the kit components have relatively long shelf lives, and of course the kit receives dedicated support from Thermo Fisher Scientific. And for more information on the availability of the new kit, I encourage you to visit the website at the address here, www.thermofisher.com/HIVDR, and you can sign up for information on when this kit will be available commercially. So with that, I'd like to thank you for your attention.
Dr. Stegan, Dr. Wood, Dr. Williams, I want to thank you for your time today and for your important research. And before we go, I want to thank our audience for joining us today as well. I'd also like to thank our sponsor, Thermo Fisher Scientific, for sponsoring today's webinar. Questions submitted today and during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand for 2 years until November 9th, 2024. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Bye-bye.